Hello, this is Jasmine Trigg, and I'm a scientist at Sartorius. Today, I'll be discussing a poster previously presented at the American Association of Immunologists conference. And the poster was titled High Throughput Quantification of Antibody-Dependent Phagocytosis using Live Cell Analysis. Phagocytosis, a specific form of endocytosis, is a critical component of both innate and adaptive immune responses. The uptake and clearance of viable tumour cells can be promoted with monoclonal antibodies via antibody-dependent cellular phagocytosis, also known as ADCP, or through the blockade of don't eat me signals, such as CD47. These mechanisms hold immunotherapeutic promise and are studied extensively in drug development. So here we have developed and validated an in vitro assay for the high throughput quantification of phagocytosis using the InKSight live cell analysis system. The InKSight phagocytosis assay combines Frodo for InKSight reagents and integrated image-based fluorescence measurements in a simple mix and read protocol. The data that I'll be showing today exemplify that live cell analysis is a powerful tool for quantitative, morphological, and functional assessment of ADCP, and this is amenable to screening for therapeutic candidates. The InKSight live cell analysis system is a fully automated phase contrast and multicolor fluorescence system that resides inside a standard cell culture incubator for optimal cell viability. The system is designed to scan plates and flasks repeatedly over time. In combination with our software, this offers fast, flexible, and powerful control for continuous live cell analysis. And this comprises image acquisition, processing, and data visualization. We also offer a suite of reagents and consumables that are non-perturbing, and these are cell labeling and reporting reagents. So phagocytosis is a process by which certain living cells are able to engulf and digest other cells or particles. Frodo reagents for InKSight phagocytosis use a pH-sensitive conjugated fluorophore that has been incorporated to enable live cell imaging of phagocytic events. The assay principle, shown here, is based on a low level of fluorescence of Frodo-labeled material in media at around pH 7.4, which, following engulfment and phagolysosome formation, results in an increase in the acidic environment, and this leads to an increase in the fluorescence intensity within the cytoplasm of the cell. Using the InKSight live cell analysis system, this increase in fluorescence can be automatically quantified over time with integrated software to segment the areas of high intensity. Additionally, the observed phagocytic signals can be confirmed using the InKSight HD phase contrast images, and these allow for direct visualization of cell engulfment in real time. For example, in the figure in the right here, Frodo-labeled apoptotic Jurkat cells were co-cultured with J7748.1 mouse macrophages at a range of target to effector ratios. Phase and fluorescent images at 20x were acquired using the InKSight system, and phagocytosis was quantified using the fluorescence segmentation mask, which is shown in the images here in blue. When we look at the time course, we can see that there's a density-dependent increase in red fluorescence following engulfment over the 24-hour time period. Tumour cells can be targeted with pro-phagocytic monoclonal antibodies, which engage host immune cells and promote engulfment and clearance from the body. One strategy for this is the blockage of don't eat me signals such as CD47, and these are present on the surface of tumour cells and enable evasion of phagocytosis. To investigate these don't eat me signals, Frodo-labeled CCRF-CEM tumour cells were treated with monoclonal antibodies targeting CD47 or IgG isotype control, and they were co-cultured with bone marrow-derived macrophages. If we look at the images here on the left, these are phase and fluorescence images for 5 microgram per mil anti-CD47, and these are acquired using the InKSight live cell analysis system. The images show an increase in fluorescence as these cells were engulfed. A kinetic increase in fluorescence was observed, with monoclonal antibody-mediated response being concentration dependent. To eliminate the possibility of anti-CD47 treatment resulting in target cell apoptosis and inducing efferocytosis, we also performed phagocytosis and cell health assays. Here, Frodo-labeled CCRF-CEM cells were treated with anti-CD47 or camptothecin, which acted as our apoptotic control, and co-cultured with J7748.1 mouse macrophages. We did this in the presence of the Inksite and Exim5 dye, and this is a cell health reagent. We found that anti-CD47 treatment induced phagocytosis without inciting apoptosis. To investigate phagocytosis of viable tumour cells via antibody-dependent cellular phagocytosis, we assessed two clinical monoclonal antibodies targeted against CD20. These were rituximab and truximab, which is a rituximab biosimilar, and these are both approved treatments for CD20 positive B-cell cancers. Frodo-labeled Ramos target cells at a range of densities from 100 to 12.5 K per well were treated with anti-CD20 monoclonal antibodies rituximab or truximab, or an IgG1 isotype control. And then they were co-cultured with bone marrow-derived macrophages. If we look at the time course on the left here, this shows that truximab-induced ADCP response is both time and target cell density dependent. The results in the middle show an increase in fluorescence for truximab and rituximab compared to IgG control at all of the target cell densities that we studied. Lastly, rituximab treatment showed a concentration-dependent increase in ADCP using the target cell density of 50 K cells per well. We also assessed the effects of anti-CD20 monoclonal antibodies in inducing phagocytosis using primary macrophages as the effector cells. Monocytes isolated from whole blood were differentiated into macrophages using cytokines. Frodo-labeled Ramos cells were treated with monoclonal antibodies targeting anti-CD20. Again, here we use rituximab or truximab, or IgG isotype control, and co-cultured these with the primary macrophages that we've differentiated. So the macrophage differentiation itself was monitored and quantified using the integrated software of the Inksite Advanced Label-Free Classification Analysis. And we can see the images of the classification masks here. So on the left, we have images for the classification mask for monocytes, which was on day 0 with a pink outline. And the right-hand image shows macrophages with a blue classification mask outline on day 7. Looking at the time course kinetic data, this shows that rituximab and truximab induced ADCP in a concentration-dependent manner, with similar kinetic profiles being observed over the 4-hour time period. Next, we investigated some native and engineered rituximab Fc mutants. Frodo-labeled Ramos cells were treated with native and engineered human rituximab isotypes, anti-CD20 IgG1, anti-CD20 IgG1 FUT, which is a non-fucosylated Fc mutant, and anti-CD20 IgG1 NQ, which is a non-glycosylated Fc mutant. And they were co-cultured with bone marrow-derived macrophages with a 1 to 2.5 K effector target ratio. If we look at the microplate graph on the left here, this shows the kinetic response for total red object area over 4 hours. We observed concentration-dependent ADCP response for anti-CD20 IgG1 monoclonal antibody and the Fc mutated IgG1 FUT, but not for the IgG1 NQ or IgG control. And the EC50 values for these were shown in the table here. These results align with previously observed effector functions. Additionally, we studied ADCP using adherent target cells that had varying HER2 antigen expression profiles. So ADCP was examined using HER2 positive AU565 or HER2 low MDA adherent target cells. Cells were labeled with Frodo and then treated with an anti-HER2 monoclonal antibody and co-cultured with bone marrow-derived macrophages. If we look at the images, anti-HER2 induced an increase in fluorescence for AU565 cells, but little to no fluorescence was observed for MDA-MB23 cells. The time course shows an ADCP response for AU565 cells and reveals that this occurs in a time and concentration dependent manner. Anti-HER2 induced ADCP for AU565 cells, but not for the MDA cells. And this is consistent with established correlations between HER2 expression and ADCP response. So to conclude, the Frodo reagents for IncuCyte in combination with the IncuCyte live cell analysis system provides an image-based fluorescence solution for visualization and quantification of phagocytosis. Our experimental findings demonstrate antibody-mediated cellular phagocytosis across a range of effector and target cell types, and they substantiate the recognized pro-phagocytic effects of monoclonal antibodies such as anti-CD47, anti-CD20, and also anti-HER2. Taken together, these data exemplify that live cell analysis is a powerful tool for quantitative, morphological, and functional assessment of ADCP, which is amenable to screening for therapeutic candidates. Thank you.